Hello everybody, and welcome back to Orgo Knows. In today's installment, we will be discussing the molecular pathogenesis of hemorrhagic fever viruses, taking a more in-depth look at arenaviruses. As listed by the Center for Disease Control, there are several hemorrhagic fever viruses noted as potential bioterrorism agents, including filoviruses such as Ebola, arenaviruses such as Lassa, and flaviviruses such as dengue. Each of these viruses causes hemorrhagic fever, as specified through their classification, and have initial clinical manifestations ranging from discomfort, vascular permeability, weakness, and irregular circulation, all of which can develop into shock and mucous membrane hemorrhage. The primary characteristic of VHF causing viruses that raises bioterrorism concerns is their uncanny ability to infect via aerosolized droplets. Previous state-sponsored biological warfare programs have been successful in aerosolizing numerous biological agents such as Bacillus anthracis, laying the groundwork for extrapolation to other highly infectious agents. Over several decades of clinical and laboratory research, a general model for the molecular pathogenesis of VHF viruses has been proposed. Following an initial exposure, whether through a break in the skin or exposure to mucous membranes, the viruses initially target the macrophages and dendritic cells. These cells support viral replication and trafficking to local lymph nodes and later full-body dissemination. Infection of macrophages initiates a so-called cytokine storm, which promotes vascular leakage and hypotension. Subsequently, the apoptosis of lymphocytes occurs, inactivating much of the innate and adaptive immune response. The inhibition of innate and adaptive immunity is furthered through the suppression of interferon response pathways as a result of improper dendritic cell maturation. This specifically inhibits activation of T cells. This general mechanism can be loosely applied to most all VHF viruses. Now, arenaviruses, which are a type of VHF virus of medical and military importance, are zoonotic Ambasense RNA viruses that occur worldwide. Ambasense refers to the coding strategy of the arenavirus genome, where the synthesis of two polypeptides occurs in opposite directions separated by a non-coding intergenic region. This intergenic region acts as a transcription termination signal for the viral RNA-dependent RNA polymerase. One genome fragment encodes the glycoprotein precursor and nucleoprotein, while the other encodes the polymerase and the ring finger protein Z. The Z protein is the counterpart to the matrix protein found in other negative sense RNA viruses. Arenaviruses are broken up into two categories, New World and Old World. Each of these categories appears to utilize a different receptor for viral entry. For Old World arenaviruses, the alpha-dystroglycan glycoprotein is utilized as a receptor. Dystroglycan is a ubiquitously expressed glycoprotein that links the cell to the extracellular matrix. On the virus, the glycoprotein 1 subunit mediates attachment to this receptor, facilitating internalization and delivery to late endosomes, which permits evasion of early endosome immune sensors. On the other hand, New World arenaviruses utilize human transferrin receptor 1 as a receptor. Once within the cell, arenaviruses have a broad cellular tropism and dissemination to the liver, spleen, adrenal organs, and other organs occurs. In a similar manner to other VHF viruses, arenaviruses target the immune machinery such that immunosuppression occurs. Specifically, the viruses target the RIG1-like helicase mitochondrial antiviral signaling response, or RLH-MAVS. Immune targeting permits escape by the viruses. The viral nucleoproteins also contribute to immunosuppression through the inhibition of interferon induction pathways. As a brief summation of the information presented, arenaviruses have four primary virulence factors. First, the glycoprotein allows for viral binding to cellular receptors and subsequent internalization. Next, the RNA-dependent RNA polymerase permits efficient viral takeover and replication. Previous work has demonstrated that small amino acid variations in the polymerase can enhance levels of intracellular viral replication. In addition, the C-terminal domain of the polymerase is a virulence determinant of some Old World arenaviruses. The mechanism by which amino acid variations promote viral replication are unknown. The third virulence factor is the nucleoprotein, which promotes inhibition of the host's innate immune response. Lastly, the Z protein mediates viral budding, regulates viral RNA synthesis, and also suppresses immune function. The Z protein can induce interferon response dysregulation. And on a final note, the RIG1-like receptor signaling disruption is crucial in the pathogenicity of arenaviruses. Those viruses that cannot disrupt the signaling pathway are not considered pathogenic. Alright, that is all the time we have for today. I hope you all enjoyed the video. Thank you for watching. Farewell.